Welcome to Lab 6, which covers the nervous system. This is a very busy lab with a lot of main objectives. And here are some of those major topics for this lab. Number one, you'll be looking at the divisions of the nervous system, that is, the central versus the peripheral nervous system. You'll be learning about the structures of a neuron and also about the structure of nerves and how those are different. You'll be taking a look at the structures of the spinal cord. You'll better understand reflexes. You'll be looking at the structures of the brain. You'll be naming both cranial nerves and peripheral nerves, as well as learning a number of muscles that are controlled by those nerves. You'll be looking at structures of the eye, and you'll be looking at structures of the ear. In addition, at the end of this lab, you'll be doing dissections of both a brain and eye specimens. So let's get started with this material. So the nervous system, pretty easy to divide the nervous system anatomically. It's broken into two main sections: the central nervous system, the CNS, and the peripheral nervous system, the PNS. Pretty simple. The central nervous system is composed of the brain and of the spinal cord. Anything that is outside of the brain or the spinal cord, that is, anything that's in the periphery, is part of the peripheral nervous system. This is going to include all those nerves that you're going to be learning. So all the nerves that you're learning, what these nerves are, or what nerves are, they are bundles of axons. Bundles of axons that are traveling distances within the body, and they are contained within a connective tissue enclosure. Also, part of the PNS are ganglia. Ganglion is singular, ganglia is plural. And ganglia are a group, a swelling, a cluster of neuron cell bodies, and you'll see these in the upcoming diagrams. Looking at this visually, the colors aren't that dramatic, but the central nervous system is shown in sort of a tan color. And again, that is the brain and the spinal cord. Anything outside of that, shown in yellow, are the nerves and the ganglia. In this diagram, you will see the ganglia are these round little clusters of nervous tissue that are found outside of the spinal cord. So let's take a look at a neuron. So the neuron is not the most numerous of the cells within the nervous tissue, but it is by far the most exciting and probably the most important for our conversations in this class. These are the cells that transmit electrical signals and that process and coordinate your nervous system. Main ideas here. You'll be seeing this again in lecture. You'll also be labeling neurons in your mastering and pre-lab assignments. The neuron is quite different from the cell, the prototypic cell that you learned about in the early chapters of the course. We think of a spherical cell. Well, the neuron is quite different from that. The cell body is what you probably think of as being the cell. It has the nucleus in the center, and it has a very dark staining nucleolus. But there are a number of processes in a neuron. And just like in the skeletal system, a process is something that sticks out. So we see all of these processes that are sticking out of this cell body. Most of those are dendrites. Now you know that dend from our vocabulary means tree-like, and ite is something that's small. So these small tree-like or branch-like structures that are sticking out of the cell body. Now the other name for the cell body is the soma, and that is how this particular image is labeling that cell body. And there is also going to be a more substantial process, just one of them, called an axon. And here you see the axon is significantly longer At the end, the axon does branch off, and even along the middle of the axon, there can be a significant branch. That would be considered an axon collateral. And the red arrow here is telling us that electrical information travels away from the soma down the axon. This image, I should add to it, the information, the electrical information regarding the dendrites, information comes in through the dendrites. So electrical information is going to enter into the cell body through the dendrites and is going to leave away through the axon. Away, axon away. Note that along the axon, and only along the axon, there are some additional cells that are protecting the axon. These additional cells, I'll put them in a different color, these additional cells are Schwann cells. Okay, they are Schwann cells, and you'll see one here, and one here, and one here. Now these are separate cells. They have their own nucleus, they have their own metabolism. They create a fatty, lipid-rich substance called myelin, and that myelin creates a sheath, a wrapping around, layer after layer after layer around the axon. In between those Schwann cells, there are small gaps, and those small gaps were named after a person, and they're called the nodes of Ranvier. So that is a capital R. Ranvier, nice French word. Okay. At the very end of the axon, there are some non-myelinated extensions. These small extensions have 3 different names by which they go. One that's labeled here, terminal arborizations. Terminal at the end. Arborizations, on Arbor Day we plant trees. So the same idea, these small branch-like extensions at the end of the axon. The other name for these, telodendria, same idea, telo at the end, dend, little trees at the end. Remember, we know telo means last or end. We saw that the last phase of telophase. And you'll also see these telodendria or terminal arborizations just referred as axon terminals. So any of those terms are used interchangeably. At the very, very tippy end of any of these extensions, there are little swollen areas called synaptic knobs. And those synaptic knobs are where chemicals are released from the neuron that initiate or sustain or even inhibit electrical signals that carry from one cell to the next. These synaptic knobs can either be interacting with a muscle, telling that muscle to contract. These synaptic knobs can release chemicals that tell a gland to secrete something, like your salivary glands to make spit, or your lacrimal glands to make tears, or maybe your pancreas to secrete some digestive enzymes. These synaptic knobs can also synapse with other neurons and send a chain reaction down a series of neurons. Remember, synapse, syn means together, APS, we learned in our vocabulary, means to fit or to fasten. So a synapse is simply where 2 things come into very close contact. They fit or they fasten together, and that is, we'll see this more as we study the nervous system and how it interacts with other cells. So that was a prototypic neuron. Neurons come in different types. One, there are sensory neurons. As the name suggests, these are neurons that pick up sensations found in your skin, found around your viscera. When you feel full, your stomach stretches. That sends a sensory signal to your brain to stop eating. When your bladder fills, it sends a signal as it stretches that you need to find the restroom. When something is hot or cold or vibrating or painful on your skin, that is picked up, those sensations are picked up by sensory neurons. The other term for that is afferent. Afferent. You know from the vocabulary that AF means toward and fare means to carry. So what we're talking about is carrying toward signals toward the central nervous system. So these sensory neurons pick up information and they send it toward the central nervous system. Now, I will, in my lectures, in my presentations, I will say afferent to really emphasize the A. But please know that this term is usually pronounced as afferent, right? Afferent. But I find that it goes by so quickly, and as we're learning, we don't hear that A clearly. So afferent. On the bottom, there are also motor neurons. Motor neurons are the ones that are also called efferent neurons. EF, going away from, carrying away. These are the neurons that carry information from the CNS, from the brain, from the spinal cord, away and back toward muscles or glands. These are the neurons that send the instructional information telling your body what to do after the brain has processed some information. Finally, in between, there are interneurons. You know from vocab that inter means between. So these are the neurons found between the incoming sensory and the outgoing motor neurons. They are found 100% completely contained within the CNS, within the brain or spinal cord. They account for the vast majority of our neurons. Here I report 90%. Other sources may say 95 or even a higher percentage of all the neurons in your body are of this type. These are the processors. These are the integrators. These are the neurons in your spinal cord that are receiving all that sensory information, making decisions about it, and then sending those instructions out through these motor neurons. So let's put these neurons together. How does this all work? Notice that on the left-hand side, this is the peripheral nervous system in green, and central nervous system is shown in blue, brain and spinal cord. So let's say you've got your finger here in your skin. You have lots of sensory neurons, sensory receptors. They're picking up vibration, touch, pain, pressure. So something is triggered in your skin. That information is going to travel through this sensory neuron. It's actually going to go into the spinal cord. So you see how we entered into the central nervous system. That neuron is going to synapse with a second neuron. This second neuron is an interneuron. These interneurons are also referred to as association neurons. It is going to process, think about that information, and then it will, if necessary, synapse with an outgoing motor neuron. Notice that now it's going to leave the spinal cord and come back to the periphery and go directly to a muscle. So here we see that motor neuron or that efferent neuron carrying that information out to what is referred to as the effector. So the effector is what is effected. It's the muscle or the gland that is receiving this information. So we see here that while we classify these neurons separately, they all work together. Additionally, neurons come in variations of shape and size. So that previous list was their function. Additionally, neurons come in different shapes or morphologies. Excuse me, the most common neuron is the multipolar neuron. These are the most common. By multipolar, what that refers to is that there are many dendrites, but only one axon. These neurons can have, we know, 10,000 plus dendrites. So imagine one cell receiving signals from 10,000, maybe even as many as 100,000 dendrites simultaneously. Obviously, this neuron is really a huge processor, right? It's receiving tons of information. The picture here is not even showing it as complex as it actually would be, right? All of those information coming into that one soma. As we go through this unit, you're going to see that the interneurons and the motor neurons are of this type. So interneurons and motor neurons are both motor polar in their morphology, in their shape. Jumping down, unipolar neurons, and you may also see them called pseudo unipolar, same thing. So pseudo unipolar or unipolar neurons. These neurons have only one process, one pole, and you'll see them oftentimes here. Here's the soma, and here's that single process coming out of that soma. That's why it's called unipolar. And much of this is axonal or axonic. And this is the morphology of your sensory neurons. So the sensory neurons that we're going to see in this chapter and in this unit are going to be unipolar in their shape. We're not going to worry about the anaxonic neurons. The bipolar neurons will maybe come across them, but it will not be a focus of this course. These bipolar neurons are found in your special senses, that is in your sense of smell, hearing, and vision. And as the name suggests, they have two poles, bipolar. One is axon and one is dendrite. So here's a table that you can fill out and use this to, what is the function, location, and to what class do they belong? Are they multipolar or unipolar? So fill this table out as a way of pulling all this information together. So we've been describing a neuron. A neuron is a cell. You have hundreds of millions, if not a billion, of these neurons in your body. But we also hear the term nerve, and I want to make sure that these two words mean something very different to you because they're very different, but they tend to get thrown together. You'll hear that neurons are also called nerve cells, and this gets confusing. So here is a diagram of a nerve. Okay, so it's a connective tissue encased structure. It is composed on the inside of axons. Okay, axons. And those axons are grouped in bundles called fascicles. So this is identical architecture to muscle. Remember, in muscle we had individual muscle cells wrapped in groups called fascicles. Same idea in a nerve. In a nerve, the outer layer of connective tissue is referred to as the epineurium. The groups of fascicles are surrounded by a layer of connective tissue referred to as the perineurium. And each and every individual axon is surrounded not only by the myelin, but also is surrounded by a thin layer of endoneurium. This is connective tissue. So it looks exactly the same. And in fact, for the same reason, those connective tissue layers provide a space for blood vessels to travel through. Remember, these axons are surrounded by Schwann cells, and these Schwann cells need their own oxygen and nutrient supply. So they must be nourished, and so these blood vessels traveling through the connective tissue layers of the nerve are vital to keep that nerve alive. We've all had our elbow on the table and felt our hand go numb or our pinky go numb. And it just turns out that the ulnar nerve comes right around the olecranon, and as you keep your elbow on the table, the blood supply to that nerve gets interrupted, and that causes a dysfunction, a numbness to that nerve. Let's take a look at histology of a nerve. So in this picture here, the nerve is going in and out of the screen. You are looking, this is just a portion of a larger nerve. You are looking at fascicles. So each of these that I'm circling is a fascicle. It is a group of axons. Okay, now another term for axons are nerve fibers. Let's make sure that makes sense to us. Back in the connective tissue conversations, a fiber was the same as a protein, right? A fiber was like a collagen fiber, an elastic fiber. In muscle, a fiber referred to the entire cell. Remember, a muscle cell was also called a muscle fiber. But in nervous tissue, when you see, I'm going to nerve N, when you hear about a fiber in nervous tissue, a nerve fiber is only a portion of the neuron. It is just the axon. So what you're seeing in a fascicle is a bundle of these nerve fibers or axons. If you look closely here, you can even see, I'm going to change colors, you can even see that these individual nerve fibers are surrounded by a white layer, and that white layer is the myelin, right? So you can see that myelin wrapped around each of those. We also see these large blood vessels, right? So again, we can see that very clearly in this histological image. From a longitudinal view, taking a look, we see now the nerve structure, and I'm going to change colors here and try to draw for you. It's a little bit abstract, but I assure you that I'm not making this up. Right through here would be one of those nerve fibers. Hard to see, but here's that white shadow. There's a Schwann cell right here, and there's another Schwann cell right here. And the reason I know that, you can see that there's some cleavage. There's an indentation, and that is a node of Ranvier. You can see another one of these nodes of Ranvier right here. You can see a few of them throughout this image. And here would be another example of a node. So here you're looking at a longitudinal view as a nerve is going from left to right. You're looking at just a portion of it. So again, when you hear neuron and nerve, make sure that you are quickly differentiating which one is referring to. Let's take a look at the spinal cord. So the spinal cord, we probably, in your mind, imagine it being bigger than it really is. The spinal cord is about a typical pencil in size, okay, in diameter. And we probably imagine it being thicker than that, bigger than that. But you've seen vertebra, you've seen images of vertebra, and your pinky would struggle to go down the vertebral foramen in most vertebrae. So it's not as big around as you probably imagine. But it is a long cylindrical structure. And another thing that is usually misunderstood is the spinal cord does not go all the way down to the sacrum. The spinal cord itself, what's called the spinal cord proper, actually ends at about L1. Okay, it goes down through the vertebral foramina down to L1. Inferior to that, there's a group of nerve roots that travel down through those vertebral foramina. I'll come back to that. Coming off from the spinal cord, there are 31 pairs of nerves. These are the spinal nerves. They arise from the regions of the spinal cord referred to as the cervical region, the thoracic region, the lumbar region, and the sacral region. The spinal cord is not a perfect thin cylinder. There are a couple of places where it enlarges, and that's in the cervical region and down to the lumbar region. Where the spinal cord itself tapers off and comes to an end, there's a pointed-like structure, sort of like the sharpened pencil, the tip of a pencil, and that is referred to as the medullary cone, or in Latin, the conus medullaris. Inferior to that conus medullaris, you have the nerve roots of nerves L2 through S5 that travel down through and resemble a horse's tail, and hence its name. Cauda, you know that term meaning tail. Equina, you may recognize, equestrian, horse. So the horse's tail. So I've already alluded to this. The spinal cord has 4 regions. Some books will also have a 5th region. They'll have a coccygeal region. That is at the very tippy end of the spinal cord. They may refer to it as a coccygeal region. There are 31 pairs of nerves, again, coming off from that spinal cord. And when those nerves come off the spinal cord in 4 distinct areas, the nerves travel as a group, and a group of nerves traveling together is referred to as a plexus. So we'll see these in a moment. Let me go ahead and name these for you, or number these for you. There are 8 nerves that come off the cervical region. There are 12 that come off the thoracic region. There are 5 that come off the lumbar, 5 that come off the sacral, and 1 that comes off the coccygeal. So that's where you get your 31 pairs of nerves. Then there are 4 regions where these nerves travel off together. In the neck, the cervical plexus, going to the arm, the brachial plexus, going down the anterior thigh and leg, the lumbar plexus, and going down the posterior thigh and leg, the sacral plexus. Some books will combine this and call this the lumbosacral plexus, but I'm going to keep it separate because I think there's a lot of benefit in keeping it as 2 separate groups of nerves. So taking a look at this image then. So again, there is the cervical nerves, right? And they're numbered C1 through C8. That's a little bit different, isn't it? Remember that there were only 7 cervical vertebrae, but there are 8 cervical nerves. So that's the one change in numbers. There are 12 thoracic vertebrae, and there are 12 thoracic nerves, T1 through T12. From the lumbar region, there are 5 lumbar, L1 through L5, and then there are sacral, S1 through S5. This particular image does show one little nerve right there, a pair of nerves. Those are the coccygeal nerves, and they might be referred to as CO or CO1. The spinal cord itself is ending right about here at the medullary cone. These nerve roots that are traveling down below that spot collectively are referred to as the cauda equina. And there is a filamentous connective tissue structure that runs down, let me change colors, that runs down through the middle of the cauda equina and helps to hold the spinal cord down to the coccyx, and that's referred to as the terminal phylum, or the phylum terminale in Latin. The spinal cord is a pretty important structure, and as such is protected by 3 distinct layers of connective tissue. The outer layer, the most durable, is the dura mater. We know from our vocab that dur means hard. It's the toughest of the layers. It's a very tough collagenous layer. Outside of that dura is a space that you may have heard of, the epidural space, right? On the dura. And there's a lot of fat and blood vessels in there. So during childbirth or other procedures where a person wants relief from pain or surgical blocks, they will go into the epidural space, inject anesthetic, and until that wears off, that person is not able to feel from that point down in their nervous system. The middle layer is a simple squamous layer, so it's not very thick, but it has a number of collagen fibers associated with it that sort of looks like a spider web, and therefore it gets its name, the arachnoid mater. Below that is a space called the subarachnoid space. This space is filled with a solution called the cerebrospinal fluid, CSF. This fluid not only goes around the outside of the spinal cord, it also travels down through the center of the spinal cord through a structure called the central canal. And this fluid also has a circulation pattern and travels up around the brain as well. And then finally, there is a very delicate layer shrink wrapped to the surface of the spinal cord called the pia mater, or just the pia. It's a very delicate layer. It's going to follow like a shrink wrap layer every groove and crevice along the spinal cord. Notice if you spell these layers from deep to superficial, it spells pad, right? This is a pad around the spinal cord. Also, we'll see around the brain. Let's take a look at these meninges, plural. And also, for the first time, you're seeing a vertebra with the spinal cord within the vertebral foramen. And so what we have been seeing up until now, this whole opening would have been the vertebral foramen. And now we see that it's filled with the spinal cord, which takes up about half of that diameter of the vertebral foramen. Let's talk first about these layers. So they're clearly marked on this diagram. So the orangey salmon colored layer, the outer layer here, is the dura. That's the toughest layer. And just above that, let me change colors. I'll make it yellow because it's already yellow, but I'll highlight it more. So here's that fatty epidural space that is outside of the dura, but still within the vertebral foramen. And that would be the area that a nurse anesthetist or an anesthesiologist would be trying to send that anesthetic, right? Bring that needle in between the vertebra and inject it in that space very delicately. Right below that is the arachnoid mater. In this diagram, it's shown as kind of a white layer. And that arachnoid mater, if you notice, looks like it has these dark fibers, right? I'll draw them here, right? And these little spiderweb looking fibers is where it gets its name. Also in this area, you're seeing all this blue. All this blue color in here is fluid, and that is referring to, I'll make it purple for now, but all this blue space would be where the CSF is. So all that blue area would be the subarachnoid space. And then finally, shrink wrap to the surface of the spinal cord, a very delicate layer referred to as the pia mater. Not the best color. Let's go with some of the more obvious. But I'm drinking in purple here, but you can see there's just a very delicate layer shrink wrapped right to the surface of the spinal cord, and that is the pia mater. When you look at the spinal cord, well, first of all, what kind of vertebra is this? Oh, darn it, it is labeled, isn't it? So that makes it easy. It's a cervical vertebra, but tell me in your mind right now, if that wasn't labeled as a cervical vertebra, how would you know that it was? I'm hearing you all think, right? I'm hearing you say, hey, here's some transverse foramina, right? And so I see those transverse foramina. This must be a cervical vertebra. I also see the split spinous process, which is sometimes present on cervical vertebra. When you look at the spinal cord, you notice that it looks like a butterfly. You see these butterfly wings, and the butterfly-shaped structure is dark matter. It's the dark matter of the spinal cord. In the nervous system, we talk about dark matter and white matter, dark and white. And so the dark matter here, actually, you'll hear it called gray matter. The gray matter here is butterfly-shaped. In the very center of that butterfly, there is a hole, and that's the central canal. Let's take another look at the spinal cord, and this gives us a different perspective, and I think maybe a little bit easier to see these three distinct layers. So here we see the dura mater. It is the toughest outer layer, the very thin arachnoid mater, underneath which there's some fluid-filled space, right? That subarachnoid space filled with fluid. And then in this particular image, the pink layer is the pia mater, shrink wrapped to the surface. Notice that there's still a very rich vascular supply traveling right along the spinal cord. So the spinal cord is protected by these layers, meningeal layers, and it's not only protecting the nervous tissue, but also the vascular supply to the spinal cord. You can see also in this image the gray butterfly, the gray matter, and the gray matter of the spinal cord is broken up into three regions called horns. Okay? So there is the dorsal horn, just the backside. There is the lateral horn, change colors. And then finally there is a ventral horn. Okay, and you'll see this labeled over and over. You'll become pros at this. The area that connects the two butterfly wings is called the gray commissure. And again, in the very center, there's a canal, the central canal. What you're seeing off to the side in yellow are the nerves. So let me also help you with your orientation. The gray butterfly that you see in the spinal cord, the wings of the butterfly always reach up and touch the back side. They always reach up and touch the posterior side of the spinal cord. They never touch the anterior side. So that will always give you your orientation. So the dorsal horn will always be the part of the gray matter that is reaching all the way up and reaching toward the back. On the sides, we see the spinal nerves. So a nerve, remember, is a bundle of axons, and here is the nerve. We see it kind of the clustering, the coming together of these two branches. This region is the dorsal root, and in the front we have the ventral root. You'll also hear posterior and anterior roots, equivalent terms. And then here's a dorsal root ganglion. I'll talk more about that in lecture, but just pointing out these structures, you'll be hearing about them as we go along. Finally, in the spinal cord, there are two deep grooves in the front. There's the anterior median fissure. A fissure is a groove. And on the posterior side, there is the posterior median sulcus. Now, by convention, a sulcus is a shallow groove, and a fissure is a deeper groove. So it just tells you that in the anterior side, the groove is deeper. However, some books will mix and match these terms. So you might see the anterior median fissure referred to as the anterior median sulcus. So don't go crazy looking for a separate structure. It's the same thing. The white matter that goes around the gray butterfly is also broken up into three areas. These areas are called columns. Another word you'll see for them, each one is a funiculus. It's a fun word, right? F-U-N. So you'll see either the ventral column or the ventral funiculus. You'll see the lateral column or the lateral funiculus. And you'll see the posterior or the dorsal column or funiculus. Running up and down through that spinal cord are clusters of axons. There's a new term here I need you to know, though, and that is that within the spinal cord, there are no nerves. Instead, inside the spinal cord, we have what is referred to as tracts. It's a nomenclature type thing. So in the peripheral nervous system, a group of neurons, axons, I should say, a group of axons is called a nerve. In the central nervous system, a group of axons is referred to as a tract. And so what we see is that going up and down the spinal cord, there are ascending tracts and descending tracts. Ascending tracts are the tracts, the axons, carrying information from the periphery and going up the spinal cord toward the brain. So those are the ascending tracts. And also in your spinal cord are descending tracts. These are carrying motor information down, which will then go out of the spinal cord and go to your muscles or to your glands. The spinal cord is the main connector between your body and the brain, but there's one job that the spinal cord does all by itself that does not include the brain, and that is the spinal cord is responsible for your reflexes. Reflexes, you know what they are. They're really quick, automatic, involuntary reactions, we usually think of them as reactions of muscles. We've all watched Law and Order type shows. A person is hooked up to a lie detector test. And maybe you're not aware of this, but there's a lot of components to that lie detector test. The examiner is asking questions, and the person being asked the questions, they're looking at his or her eyes, looking for changes in the pupil diameter. They have a monitor on their skin looking for changes in sweat. They have a monitor on their heart rate, seeing if their heart rate goes up or their breathing rate goes up. All of those things typically will be increased if you're lying, right? Your heart rate goes up, your breathing rate goes up, you sweat a little bit more, your pupils change. And so these are all reflexive responses. As an example, another example that you're probably very familiar with is touching a stove or stubbing your toe. So let's talk about touching the stove. You touch the stove. Even before you are consciously aware that you are burning yourself, you will, quote, reflexively pull back from that hot surface. You'll pull back before you're even aware of it, right? You pull back and then you go, oh man, that hurts, right? You pull back before you're even aware of it consciously. How did that happen? This is the wiring, what's called the reflex arc of the reflexes. So something, in this case, a pin, poked your skin. Now you've got nerve endings in your skin. They picked up on that pain sensation. Those signals traveled through that nerve, entered into your spinal cord. Notice as information comes into the spinal cord, it always goes in the posterior root or the dorsal root. And that neuron is now going to speak with some interneurons found in the spinal cord. These interneurons are going to make the decision, hey, that's not good. And they're going to send information out through a motor neuron, always out the anterior root, and in this case, going to a muscle. So if you were reaching out, touching the stove, perhaps that reflex is telling your biceps brachii or your brachialis to flex your elbow away from that painful sensation. Again, none of this includes the brain. This is happening before that. Now, right after you pull away, your brain does become aware of it, and you become aware that you have burned yourself and all of that goes on. But the immediate response of a reflex, here are the 5 steps, and it does not involve your brain. Let's look at another reflex. This is so cool. You're walking along, and with your right foot, your bare foot, it's summer it looks like, and you step on something sharp. So those pain receptors in your right foot are going to send that signal up to your spinal cord. Notice it comes in the back door and goes in. It's going to speak to those interneurons, and then those interneurons are going to say, hey, dummy, you just hurt yourself. Go out the front side, go back down. This motor neuron is now going to go down to, I wonder what muscle would cause you to flex at the knee. That might be the hamstring group, right? And maybe your psoas major was involved in retracting that leg up. And you would do that. But what's so cool, not only did you lift up your right leg, but other interneurons in your spinal cord went to the other side of the spinal cord, a contralateral relationship, spoke to other motor neurons that now went down to your left leg and told your left leg to extend. So that would be, for example, your quadriceps muscles, right? Would be involved in causing your leg to extend at the knee. So this is a more involved reflex, but still, all of that would happen even before you were consciously aware. You would withdraw the foot from the pain, and simultaneously you would extend the other foot so that you don't fall over. Very cool stuff. And because there's quite a few synapses happening here in the spinal cord, this is referred to as a polysynaptic reflex. So just looking a little more carefully at the spinal cord, again, now you see the nerves are numbered, C1 through C8, T1 through T12, the lumbar and the sacral nerves. And then this particular image also has the plexuses labeled. So the cervical plexus, the brachial plexus going off to the arm, the lumbar plexus, and the sacral plexus. Note the sacral plexus is heading behind, going down through that sciatic notch and heading down the posterior thigh, whereas, it's hard to see here, I know, but those lumbar nerves are traveling down the anterior side of the pelvis and are traveling down the anterior thigh. As you look at the cadaveric image here, you appreciate that the spinal cord, you can see those the nerve roots merging to make the root, and we'll be seeing more of these images. You're going to be learning the names of quite a few nerves. Now, the great thing about naming nerves is that you already know most of these terms. These terms, the nerves are named either for the region of the body through which they pass, or they're named for the muscle or the region that they go to. So the naming of, or the bone along which they travel. So the naming of nerves should not be a challenge for you. You'll have to take out your book, make some notes, but I think you'll find this to be pretty straightforward. So for example, the nerves that go over to the arm, the brachial plexus, there are 5 nerves that run down the arm. There's the axillary nerve. Makes sense. It's up in the armpit area. There's the radial nerve. We're going to see that it comes down and runs along the radius. It makes sense. The ulnar nerve comes down, runs along the pinky side. Again, runs along the ulna. That makes sense. There's a nerve that goes right down the middle of all that, and that is the median nerve. Okay, right down the middle. And then finally, there's a nerve referred to as the musculocutaneous, which is not shown on this image. Now, there are 20-some muscle nerve combos that you're going to be learning. For example, there's a separate handout for this. But you're going to learn that the deltoid muscle is controlled by the axillary nerve. Now, folks, this is not that hard. You have to do some matching up. But if the deltoid muscle is sitting in the shoulder area, as you know, and the axillary nerve is sitting here in this area, I would argue that that is not a huge stretch, right, for you to learn that the deltoid muscle, right, that would be sitting here is controlled by this axillary nerve. Likewise, the radial nerve, right, the radial nerve comes down and it goes right here, right, and it's going to control your triceps brachii. The muscle that's, or the nerve that's not shown here, the musculocutaneous, is going to be controlling your biceps brachii. So look at that handout. I think there's 26 or 27 of those nerve muscle combinations that you'll need to learn. So while we're not directly testing on the muscles and bones from the first lab exam, you can't forget it. You'll be applying that information as we move forward. Let's take a look at the brain. The brain is the most complex of the organs that we have studied so far. You're going to learn quite a bit about the brain. This slide is showing you what's called the functional areas of the brain. That is, each lobe of the brain has a number of jobs that it is primarily responsible for. There are certain regions of the brain, when damaged, the person can no longer do certain things. So if you were to lose a significant portion of your frontal lobe due to a stroke or to some sort of brain injury, you would have deficiencies in your thinking, your memory, your behavior, and it's your frontal lobe that initiates your movement. So when you say, or when your brain tells your biceps brachii to move, those instructions are coming from a portion of the frontal lobe. If instead you had damage to your parietal lobes, left and right, then you would probably have some issues with language, and you would also have issues with touch and other sensory type of information. If you had damage to your occipital lobe, that would affect your sight, perhaps. If you had damage to your temporal lobe, you could have significant issues with your hearing. Also some learning, some feelings as well. There is a lobe that's not shown here, but if you were to crack open this brain along this sulcus, this fissure here, and look inside. There's another lobe called the insula. That's where your taste centers are. If you were to have brain damage to your brain stem, this purpley area, then you would have issues with your ability to breathe or regulate your heart rate or regulate your body temperature. And if instead you had issues or damage to your cerebellum, you would probably have issues with your balance and coordination. Now, there's a whole lot more that the brain does, but these are the main areas, the main jobs, if you will, of these regions of the brain. As you look at the brain in a mid-sagittal cut like this, there is a number of structures that you'll be looking at. You will be naming the lobes, right? The frontal lobe, parietal lobe. You'll be looking at the occipital lobe. Temporal lobe would be on the far side right here. When you open it up mid-sagittally, there's a very thick band of white matter here. That is the corpus callosum. There's another thick band of white matter on the bottom side. That's referred to as the fornix. There is an area here where I'm circling. This area is the thalamus. Right below that is the hypothalamus. So we have the hypothalamus right below the thalamus. There's a large oval region in the brainstem referred to as the pons. There's also an oblong region called the medulla oblongata through here. And then if you imagine, here's the occipital bone coming around the base of the skull. Everything that passes through that foramen magnum would be spinal cord. There's a few more structures on here that you'll be learning, but those are some of the big ones for now. In addition to learning a number of the nerves that come off the spinal cord, the spinal nerves, there are also 12 pairs of nerves that come off the brain and the brain stem, referred to as the cranial nerves. These nerves are different in that they are numbered when they're either named with a name or they are numbered always using Roman numerals. And you'll see those Roman numerals here, 1 through 12. So if you don't know your Roman numerals, it's time to learn them. And specifically in this lab, you will be seeing the optic nerve coming off the eye in your dissection. And you're going to see the vestibular cochlear nerve in the ear as you learn about the ear. Now, you are not responsible for identifying these nerves on the brain. So if you flip over a brain model or look at your atlas and your book, you're going to see these cranial nerves labeled. You are not responsible for identifying the cranial nerves, but you do need to learn the cranial nerves, what they do, okay? And in some of these cases, some of these cranial nerves also control muscles. So for example, the accessory nerve, the 11th cranial nerve, you're going to learn, controls your trapezius and your sternocleidomastoid muscles. Think about your shoulder muscles. The ability to shrug and turn your head is controlled by one of these pairs of cranial nerves, the accessory nerve or cranial nerve 11. You'll also be learning that a number of muscles in your face, for example, the zygomaticus, the orbicularis oculi, the orbicularis oris, are controlled by the facial nerve. And you'll be learning that the muscles that move the eye are controlled by the ocular motor, the trochlear, and the abducens nerve. So that information is coming, and you'll be learning more about that in this unit as well as in lecture. 2 more structures to be looking at today, structures of the eye, and then in the next slide, the structures of the ear. You'll also be dissecting an eye today. Just looking at this quickly, and then you'll be digging in a little bit deeper. But there are 3 main layers of the eye. The white of the eye is called the sclera. Now, vocabulary, we'll learn later that sclera means hard. As you are cutting through this outer layer, you will experience just how tough and hard that outer layer is. Not hard like a rock, but very, very tough. The middle layer is referred to as the choroid. The inner layer is the retina. Retina, you know reticulum You've seen the endoplasmic reticulum. You know that retina, or reticulum means a network, reticular fibers. And so the retina is a network of receptors that receive visual signals, right? Visual receptors in the eye. The optic nerve, there it is. That is cranial nerve number 2. That's coming off the posterior eye. It's going to carry that visual information from the retina to the occipital lobe of your brain, as well as other parts of your brain. We see some of these muscles of the eye coming around. You will be learning, and did already learn, the muscles around the eye. This would be the superior rectus, and this would be the inferior rectus muscle around the eye. The lens of the eye is shown here. In your body, in a freshly killed specimen, it would be a clear, very crystal clear structure. In your dissected eye, it's going to be much more cloudy, kind of yellowish in color, simply because it has been sitting in the solution. The outer layer covering where the light goes in is called the cornea. You may have scratched your cornea before. You know that that's a very, very highly innervated, very painful scratch to scratch the cornea. In the front of the eye, there's a space, an area referred to as the anterior cavity. And in that anterior cavity, there's a liquid called aqueous humor. In the back of the eye, in the posterior cavity, there's a jelly-like substance called the vitreous humor. So in lecture, we'll talk about how this all works together, but your job now is to learn the structures of the eye. And then finally, the ear. Again, focusing on the structures, not the physiology, not how this works. But the ear is broken up into 3 areas. There's the outer ear, there's the middle ear, and there's the inner ear. The outer ear is composed of the pinna. You may also see this called the auricle. Right, that's the flappy part of your ear. And I guess we'll talk a little bit about how the ear works. So the sound comes in through your canal, ear canal, and hits this membrane. Okay, you know it as your eardrum, but we're not going to give credit for that. We're going to call it the tympanic membrane, like the timpani in the orchestra, the big drum. So that tympanic membrane is going to vibrate. That vibration is now going to be transferring that vibrational energy to 3 bones. These 3 bones collectively are called the ossicles. The first one is the malleus, then the incus, then the stapes. You'll also hear stapes, so stapes or stapes. These now are the last bones that you need to learn. So in the skeletal unit, we learned 200 bones. Of the 206 bones, there are 3 of these ossicles in each ear. So now you know all 206 bones in your body. That vibrational energy is going to go on the stapes, and there's an opening behind the stapes. And that energy is now going to come into the entryway here. This entryway is called the vestibule. The vestibule, you may know that vestibule just means an opening into something. Architects talk about a vestibule in a building or in a church. In your body, you have a nasal vestibule, an oral vestibule. There's a vaginal vestibule. And as you come into the opening of the inner ear, you look off to one side, you see 3 semicircular canals. These 3 semicircular canals are involved with balance. To the other side, you now see this spiral structure called the cochlea, and that's where your hearing is occurring. We'll deal with more details later. The information for the semicircular canals travels through a portion of a nerve called the vestibular nerve. The information for hearing travels through the cochlear nerve. These 2 nerves come together. So these 2 nerves come together to form one nerve, and that is the vestibular cochlear nerve, and it is also your cranial nerve 8. Okay, so you'll see it call it either the vestibular cochlear nerve or vestibular or the cranial nerve number 8. So at the end of this lab, you're going to be doing a couple of dissections. Each of you has a sheep brain. The sheep brain is going to have, hopefully, the outer layer on here. That's the dura mater. Here is some spinal cord. Keep in mind that in a four-legged animal, their architecture is a little bit different. So in our brain, the cerebellum would be sort of down below the lobes, but on a sheep, the cerebellum is here, right, and the occipital lobe would be deep to that. You're also going to be having an eye, and you'll be cutting the eye. You'll be doing some labeling, some activities. You have a choice. You can either, after you've done your dissections, lay out your pieces on a piece of paper, write the structure and draw a line to it the best you can, or you can take a video of pointing to each of the structures on the dissection rubric list and upload that. So you'll be either uploading images, labeled images, or uploading a video to your instructor at the end of this lab.